Hello, friends. Welcome to Crack Defense. So, in this chapter, in this class of ours, we will cover blood. Okay, we'll study everything about blood. So, what blood is a connective tissue, and in human, blood is an extracellular fluid. And in case of bacteria, blood is an intracellular fluid. Now, what is an extracellular and what is an intracellular fluid? So extracellular fluid means it doesn't go inside the cell. Okay, it is present outside. So in case of human, we have extracellular fluid. Okay, blood doesn't go inside the cell. Blood is present outside the cell. So this is blood. Blood is present outside the cell. It will not go inside the cell. But in case of bacteria, blood is intracellular. Okay, that is present inside the cell. Now we'll study different components of cell. Okay, so our cell, our blood, it consists of plasma and blood cells. Okay, so we have both. We have plasma also, we have blood also. Plasma constitutes 55% of the blood, and rest 45% are blood cells. So in these blood cells, we have WBC, we have RBC, and we have platelets. Okay, RBC is 45%. So if you see this diagram, okay, here this is the test tube. So when you go to the doctor and you want to have any kind of test, they will take your blood sample and they will collect it in a test tube. Okay, so after some time what you'll absorb is that these blood cells will settle down because they are more denser, and the plasma will settle here. Okay, because plasma is less dense when we compare that with cells. And then we'll see here, you can see how much of red blood cell are there. Okay, and then you see this white space. Okay, these are white blood cells, and then we have plasma. So you can see the percentage of white blood cells and platelets when you compare them with red blood cells. Okay, so we have more of red blood cells in our body. Then these are plasma. And when we talk about the technical name for RBC, WBC, and platelets, so RBC are called as erythrocytes. They are also called as erythrocytes. WBC are called as leukocytes, and platelets are called as thrombocytes. Okay, so these are the. So if we revise this, so RBC, erythrocytes, WBC, leukocytes, and platelets, thrombocytes. So you should remember these names because again, these can be asked. Then if we talk about plasma, so plasma consists of ions, water, proteins, nutrients, waste, and gases. We'll study about plasma in detail also. So this is the slide where these are the components of plasma. Plasma consists of water. So 90% of water is present in plasma. Then we have plasma proteins. These proteins are albumins, globulins. Okay, so these are plasma proteins. Then we have electrolytes. So here in place of electrolytes, we have electrolytes like Na plus, Cl minus ions. Okay, they are the major quantities in plasma. Then here this diagram is given, and here they are showing that this is plasma, the yellow fluid. Then these are the other components in plasma. These are nutrients, hormones. Hormones are also they also reach different part of our body with the help of plasma. In our coming lessons, we'll also talk about endocrine system. So there we'll discuss about hormones. Okay, and these hormones, they travel through blood. Okay. Then waste and blood gases. So these are the components of plasma. Now, they are the major types of plasma proteins. So, we have albumin, it constitutes 60% of plasma. Then we have globulins, then we have fibrinogens. Okay, albumin is the major component of osmotic pressure of plasma. Osmotic, or it helps in the osmosis process. Okay, osmosis means fusion of from one membrane to another membrane. So, that is what albumin helps in. Then we have globulin. Okay, so these are antibodies and they transport proteins. They are also called as immunoglobulins. Then, if we talk about fibrinogens, so they are 4% in our plasma, and they function in blood clotting. So, we will study about this clotting thing also in our coming lessons. Okay, so stay tuned for that lesson also. And then these are some other factors that are present in our plasma. Now we'll study the formation of blood. Okay, so where blood is formed. So blood is formed in the bone marrow. Okay, this is the bone marrow. So in our bone marrow, bloods are formed. So we have red blood cells, we have white blood cells, and we have platelets. So in blood cells, these are the types. We have lymphocyte, monocyte, eosinophils, basophils, and neutrophils. So these are the types of white blood cells. We'll discuss about each of them. Now here, first we'll come with our RBCs. We'll talk about our RBCs. Okay, they are the major constituents in blood. Okay, and the shape of RBC is concave. You can see here they are biconcave, they are denucleated, they are elastic, they lack cell organelles like endoplasmic reticulum and ribosomes. I hope by now you know these terms. Endoplasmic reticulum and ribosomes, their functions. Okay, and RBCs are red in color, WBCs are white in color. RBCs are red in color because of the presence of hemoglobin. And then we have function, so they transport oxygen and carbon dioxide. And the life of RBCs are 120 days. After 120 days, they are getting destroyed in the spleen, and again, new set of RBCs will be formed in the bone marrow. And when we have less amount of RBCs in our body, it can cause us anemia. Okay, anemia is caused because of deficiency of RBCs. So I hope this much is clear. And also RBCs, when they carry oxygen, they are also called as oxyhemoglobins. So we have hemoglobin and we have oxyhemoglobin. When RBC carry oxygen, they are called as oxyhemoglobins. Now we have these leukocytes. So leukocytes are colorless, and they are rounded or irregular. They are nucleated. Now these are also formed in bone marrows, lymph node, spleen, and thymus. The function is they act like soldiers or they help in healing. So these are the types of WBCs, and these are the percentage of WBCs. So neutrophils, these are 45 to 75%. So this is the major constituent in WBCs. Then we have lymphocyte, it is 20 to 40%. And then eosinophils, monocytes, and basophils are present in very small quantities in our blood. Now we have this table where we'll study about these neutrophils, eosinophils, basophils, monocytes, their functions. Okay, so before that, here it is written that these are the types of WBCs. Okay, so here it is written granulocytes and agranulocytes. So they have written neutrophils are granulocytes. Okay, the eosinophil is also granulocyte, monocyte is agranulocyte. Now what does this granulocyte and agranulocyte means? Okay, so granulocyte means presence of granules, and agranulocyte means no granules will be there. And how will we remember this? So here you can notice that all these WBCs which are ending with "phil," these are granulocytes. Okay, and here monocyte, and then we have lymphocyte also. Okay, then we have lymphocyte. In lymphocyte also, lymphocyte and monocyte, they end with "cyte." Okay, cyte is the suffix. So monocyte and lymphocytes are agranulocytes. Here if we see the diagram, so here you can see that neutrophils, eosinophils, and basophils, you can see the presence of these small dots. Okay, these are granules. Here also you can see these dots. These are granules. Here also you can see these dots. These are granules. But in case of lymphocyte and monocyte, these granules are absent. So in this diagram also you can notice this. Then we'll read the functions. So neutrophils, they defend against bacterial and fungal infections. They make up 60% of total leukocyte control, and the lifespan is about 5 days. And if we talk about eosinophils, so they are inflammatory cell and they respond to allergic actions. Then we have basophils. Okay, so basophils, here in basophils, they are responsible for allergic actions and they release histamine, which causes vasodilation. Now, what is vasodilation? Vasodilation means they will cause your blood vessels to dilate. Okay, and heparin or histamine, they helps in broadening of vessels. That is called vasodilation. Then we have monocyte. So monocytes are microorganism eaters. Okay, so they are called as microorganisms eater. They eat the unwanted microorganisms that has entered your blood. And the process through which they eat, that is called phagocytosis. So phagocytosis is the process through which monocytes, they eat microorganisms. Okay, and if we define phagocytosis, so what phagocytosis is, it is the process by which a cell uses its plasma membrane to engulf a large particle. Okay, so if we visit our next slide, so here they have given here this is the phagocytosis. Phagocytosis means intracellular killing. So here you can see that this is the phagosomes and the lysosome that is present in the blood. And we have already studied in our previous classes that lysosome that are present in the blood, they help in killing of unwanted microorganisms. So this will react with this phagosome and phagolysosome will be formed, and then the unwanted bacteria that has entered, that will get digested. They will kill the unwanted microorganism. So this is the process of phagocytosis through which intracellular killing takes place. Then we have other WBC, that is lymphocyte. So this is lymphocyte. Okay, this is lymphocyte. Lymphocytes are of two types. They are B lymphocytes and T lymphocytes. So we have two types of lymphocytes, B and T lymphocytes. B lymphocytes, they attack invaders outside the cell, and T lymphocytes, they attack invaders inside the cell. So we'll see their function here in this slide. So here in B lymphocyte, it was written that they attack invaders outside the cell. So how they attack invaders outside the cell? So this is the diagram. Here you can see that B cells, they release neutralizing antibodies. Okay, so antibodies are present on the wall of these cells. These are antibodies. So when an unwanted virus has entered our body, this B cells will release these antibodies, and these antibodies will get attached here with this cell. Okay, and they will protect the cell. They will form receptors, they will protect the cell. And now, because these antibodies have taken the space, now virus cannot enter the space. Okay, these are the entry channels through which virus can enter. So these antigens, they will block these entry cell, and because of which the unwanted virus will not be able to enter our cell and they'll not be able to infect our cell. So this is the way through which B cells keep us immune. Okay, they protect us from unwanted microorganisms attacking us. And then if we talk about T lymphocytes, so here in case of T lymphocytes, it was written that T lymphocytes are, they attack inside the cell. So how they attack inside the cell? If we see, suppose here by chance a virus or a bacteria has entered the cell. Okay, suppose it has entered the cell. So now what will happen? Now this cell got infected. Okay, now This, if this is infected, it can it can spread the infection to other cells also. So what T cells will do at that point of time, T cells T cells will recognize the infected cell, okay, and then they will cause these cell to die. They will release the viral antigen, and then they will cause this infected cell to die, and through which they will protect us. They will protect our cell. So this is the process through which B lymphocytes and T lymphocytes react with the unwanted microorganisms, and they keep us safe. Okay, so this was the process. Now in the next class we'll study this. We'll study about platelets that was left. So we'll meet there. Till then, take care and goodbye.